Hi, everyone. It's a pleasure to be here and to be presenting on our updates on the computational tools that have been developed at our group at Translational Imaging Research Unit at the 3B Center at CHOP. My name is Anahita Fathi Kazerooni, and I'm an instructor at Department of Neurosurgery, UPenn, and Division of Neurosurgery at CHOP. So I'm going to jump right into the problem statement. Tomorrow I'm going to talk a little bit about the details of AI research, but today I'm just going to discuss the most important parts that relate to image processing, as was discussed by my colleagues as well. So AI research in any fields, including medical imaging and specifically radiomic research, needs big data. And big data needs data preparation and processing in a systematic and standardized way so that they can reliably be passed through machine learning and deep learning algorithms. In the context of adult brain tumors, there have been many studies at different centers working on building pre-processing pipelines for brain tumor scans, and some of them have become available in some openly accessible software packages, like one of them that was developed by my group at my previous group at Penn, which is called BRAT pipeline, which is incorporated with CapTK software. And what it does is that if you pass through DICOM images, it will convert it to NIfTY format and then reorients the images to standard radiology space, and then co-register all of the images with each other and with a reference atlas. And then they will do the, sorry, just to make sure that I can, then there will be a brain tissue extraction, or the so-called skull stripping method that will separate brain tissue from the rest of the structures, and then tumor subregion segmentation, which will facilitate radiomic feature extraction or passing the images through deep learning models. I'm going to focus on two core steps for any brain tumor pre-processing pipeline, which are brain extraction or skull stripping and tumor subregion segmentation. So brain tumor segmentation, why is it important? Because it helps in tumor detection and diagnosis. It facilitates radiomic studies, and it can help in pre-op treatment planning. For example, what is the extent of tumor invasion, or what is the volume of enhancing tumor? It can help in volumetric assessments for monitoring tumor progression, for example, based on RAPNO criteria. And brain extraction or skull stripping is important because it can help in tumor segmentation. It can also help to standardize different images across different centers with each other. It also helps in the identification of the patients because it removes the bony area and the skin from the soft tissue, and therefore the face of the patient cannot be reconstructed. It also helps in standardized feature extraction as well. So one of the problems that we have when we are applying the models that are developed on adult brain tumors to pediatric cases is that pediatric brain is actually developing. So across different age groups, for example, as you can see from this figure, from a 3-month-old to 1-year-old to 5-year and 18-year-old, the brain structure is changing constantly. Also, the signal intensities on MRI images also are changing. So it is difficult for the models that are trained on adult scans to find the patterns in pediatric brain tumors, brain tumor scans. So one of the challenges of skull stripping methods, for example, is that in younger children, as you can see here, The bony structure or the skull has not fully developed, and therefore we cannot find the desired T2 signal decay on T2 MRI scans, which is something that usually the skull stripping methods are based on. Another challenge of applying adult models to pediatric scans is that there's a difference between subregions of interest in pediatric brain tumors. For example, in adult brain scans, usually it is very well accepted that the enhancing tumor should be separated from necrosis, from a peritumoral edema, which includes tumor invasion as well. But in pediatric brain tumors, based on RAPNO criteria, it is recommended to separate enhancing tumor from non-enhancing tumor, cystic core, and edema for volumetric measurements and response assessment. And other challenges that we have is that, well, if we do not want to use adult models and just want to use pediatric models, the problem that we have in development of such models is that brain tumors in children, it has a lower incidence than their adult counterparts, which is good, but in order to be able to build models that are generalizable and can be used for any future patients, we need more data. And there's a lack of multi-center data at the moment, and some of the imaging protocols have not been standardized. So we have come across a lot of scans that even T1 pre didn't match T1 post contrast. And therefore, we had to exclude those patients from our cohort, basically. Also, there is more heterogeneity existing in pediatric brain tumors. So what we did was we took the models that were developed on adult brain tumors and used transfer learning to transfer the techniques to a large cohort of pediatric brain tumors that was collected at our group, and we developed deep learning models for brain extraction and tumor subregion segmentation that I would like to call it multi-parametric, multi-scanner, multi-histology, multi-center models. And here I have just showcased two examples of patients who are 2-year-old, 18-year-old, and you can see the prediction of our models for creating brain mass compared to the expert segmentation and tumor segmentation based on our model compared to expert segmentation. So our cohort that was collected, actually, we went back and collected retrospectively something over 350 patients, but not all of them were useful, as I mentioned. So we finally had a cohort of something around 250 patients from internal cohort, which is from CHOP, and external cohorts from a few other centers. The MRI scans were clinically acquired, and we focused on standard multiparametric MRI scans that are mostly accessible at any center, including T1 pre and post, T2 and FLAIR scans. These MRI scans were acquired across different scanners with different field strengths and with different vendors, including Siemens, GE, Philips, and Toshiba. And our cohort also included multiple histologies like low-grade glioma, medulloblastoma, high-grade astrocytoma, DIPGs, and ependymoma. And all of these images were collected, processed, prepared, and modeled by our amazing team at Translational Imaging Research Group, which is a multidisciplinary team of people with different backgrounds, researchers, data scientists, computer scientists, and neuroradiologists. And for a lot of these patients that we collected, the neuroradiologists had to go back and segment some of the patients multiple times to reach a consensus among each other. So the results of our skull stripping model is summarized here. As you can see, based on DICE score, which shows the model performance, on the validation set, we achieved a high DICE score, which was reproduced on the internal test set. And the internal test set was kept unseen during model training phase. And then on the external test set, which was from external sites other than CHOP, we see high generalizability of our model. And here I have shown a few examples of two methods for skull stripping that are developed for adult brain tumors across different age groups. And they usually tend to undersegment or oversegment the brain tissue in pediatric brain tumors. But our model consistently is performing accurately for patients with different ages. And our tumor segmentation model also showed a high performance on whole tumor segmentation on the validation set, which was reproduced on internal test set and generalized on the external test set. For enhancing tumor and non-enhancing core, we had slightly lower but still highly accurate model. As you can see from these plots, our whole tumor segmentation consistently works well across different patients. The enhancing tumor and non-enhancing core are sometimes difficult in some patients if the quality of MRI is not high, or there are weakly enhancing tumor regions that may be segmented as the other group, which is usually very difficult for expert neuroradiologists as well to segment. And here you can see two examples of the patients from our internal test and external test sets. So the data from these patients were completely unseen by the model during the training phase. And this is the segmentation by our model. This is the expert segmentation. As you can see, the whole tumor dice is very high for both enhancing tumor and non-enhancing core also are highly accurate. As Mariam was mentioning in her talk, there are several features that the radiologists usually use. They're called Vasari features. And we show here that our prediction of, for example, enhancing tumor volume compared to the expert or manual segmentation by the neuroradiologist has a high agreement based on the correlation plot that you can see here. And similarly, for the non-enhancing tumor, cystic component, and edema. So there have been just a few other studies working on specifically pediatric brain tumor segmentation. And even though they have built the base for other researchers and other studies like ours, but usually they didn't use all of the MRI scans that are required for being able to differentiate the tumor subregions from each other, or their cohort didn't have all of the histologies that our cohort had. Also, our data basically was a large cohort of pediatric patients carefully chosen. And the regions that were segmented were RAPNO-based or RAPNO recommended. And we show that our model seems to be performing a little better than some and a lot better than some others in segmentation of brain tumors in pediatric cases. So in conclusion, even though it is much needed and everybody admits, application of AI in pediatric brain tumors is necessary, but it is limited because mostly we don't have enough data. Some of it is because of lower incidence, which is good. In general, we don't want any children to have brain tumors, but to be able to cure the future patients, we need data, retrospective data, to build our models based on those. So if we have more standardized acquisition protocols across the centers, if we have more contributing centers, our modeling would be enhanced. And well, there's a large heterogeneity across pediatric brain tumors, which is inevitable. As I showed, AI research needs process data which is adapted to pediatric brain tumors. And here I showed two examples of the key steps in image processing for pediatric cases, which is brain tumor subregion segmentation and skull stripping. Next, we are updating our pediatric brain tumor pre-processing pipeline, and we will make it accessible for the community. And our models are going to be extended to include post-op images as well. That could be helpful for treatment surveillance and assessment. I would like to acknowledge everybody who has helped in gathering all of the data and information and facilitating the research at our lab, specifically our team members at Translational Imaging Research Unit. Thank you.